And so, in fact, our first speaker is connected to this hall, the Yoshimori Osumi Hall. Dr. Jürgen Kriel has spent his first research career part in South Africa, looking at autophagy and in fact termed lethal autophagic flux in his research. Jürgen is an early career imaging scientist in advanced bioimaging. He managed the core facility, EM core facility in South Africa. In fact, he has set up the first volume EM and correlative workflows. And he is now a senior research officer at WEHI, the Brain Cancer Research Lab in Australia, focusing on multimodal imaging and spatial omics. So very exciting, Jürgen, and we look forward to your talk.
Thanks, Ben. Hi, everyone. Thanks for that lovely introduction, and obrigado gozaimasu to the organizers for inviting me to speak today. Yara gave me a slightly confusing invite asking, share your experience from moving from one institute to the next. A lot of people change jobs, Yara, but what I think I'll do is share my journey going from an imaging-focused institute to a bioinformatics-focused environment, and end off with how we can better help smaller facilities adapt to the standards set by larger institutes. So even though I make reference to two institutes, at the end, it's basically between small and large facilities. I'm not really picking sides, even though Stellenbosch is by far more beautiful than Melbourne. And I do make some gross oversimplifications, so don't take offense on that and walk out. So A Tale of Two Cities, it was the best of times. Ben mentioned we did a lot of autophagic work and a lot of claim work, and we set up a serial block face lab at a new imaging or at a new biomedical research institute at Stellenbosch, and we set up a claim workflow to identify autophagosomes more accurately. Also took our claim workflow one step further, and in collaboration with members from the CRIC as part of the CRIC Africa network, so thanks to Lucy and this is work by Caroline Beltran, we did volumetric claim on cleared lung tissue, TB-infected lung tissue, and what we could show was that even though tissue clearing does affect the membrane quality, it was still good enough to segment out larger structures within this block, and we could identify subvolumes in a lesion that wasn't as apparent in micro-CT. So combining light sheet, CT, and volume EM with these different modalities. It is still the best of times. If you've read the book, it changes topics. But yeah, it's still the best of times. So I've transferred the learning I did from aligning imaging data sets to now aligning spatial transcriptomics data sets. This preprint came out this week, and I got notification that we submitted it for review today. And I'm calling this SMINT, which is Spatial Multi-Omics Integration. So what we're taking is spatial transcriptomics, spatial metabolomics, and serial aligning them. So basically omics tomography, you can think about it like that. And at the end of the day, what I'm busy with now is setting up a little 3D viewer for multiple samples so that we can see our different cell types this is a astrocytoma sample from a patient, so that we can see cell type distribution in 3D and knowing exactly what they are in the tumor microenvironment. And I also CT scan the samples beforehand because I love 3D, and then we can make the alignment more accurate according to that. But a part of me did die inside, so when we started to do our segmentations, I thought, all right, I got this. I've done segmentation before. And compared to what we did or what we got from the manufacturer here, which is just nuclear dilation, I thought, okay, I can do better than that. So train a cell post model to segment the structures more finely, and that worked out quite well. Like, okay, great, this is going to make our data better, right? Well, not really. So what we found was if we just segmented the nuclei and assigned transcripts to that, the overlap between that and whole cell annotation was about 87%. So that was close enough for a large volume of samples. And the electron microscopist in me was very offended because we're not using the whole cell segmentations anymore. But we got a good enough amount of data for 3D alignment out of that. So we're managing this data at WEI. We have all our instruments connected to a local server, as most do, and the raw data is regularly backed up with tape. We do our computation on our cluster called Molten. This is our mascot, Molten, the compute monster. And all our processing gets done on a vast scratch space. So vast is like extra board compute space. And then here we play around with all our different processing steps until we get the final product process data that then gets fed back, and that's backed up. So this is the concept of read once, write once, read many, worm. I know I have a lot of silly pictures. But yeah, you write it once, read it many times, and then push it back. What I've also learned along this journey is working with objects rather than just images themselves. So in one of our ZAR files, and shout out to yesterday's demonstration where we could add, well, we could make suggestions for what we want to add to ZAR, this is what I work with currently, is one ZAR file has my images, my labels, my transcripts, the shapes, and then an AND data table with all the gene information associated with that. And then I align all these different ZAR files to give me a unified combination of this, what we call unified clustering between spatial transcriptomics and metabolomics. And this is now the data sets we're working with rather than just, well, just images, because images are also quite massive. And this is getting out of hand since we're trying to also develop a centralized research data library for brain cancer research across Australia. So image data actually makes up a very small, like a little component of this entire map that we're trying to build. And if we had a unified file format to only push these into for a centralized repository, that would make life much easier, because on the clinical side, we have MRI data, and on the laboratory research side, we have various kinds of imaging data and spatial proteomics data sets that we're trying to integrate into one large centralized repository. So then looking back, when I'm comparing focused research institutes between smaller core facilities, there's some big differences, but these two kind of compete under the same rules, right? So if you have scattered facilities, this is everything consolidated in one location. Sometimes you have a broad focus, so you would have geology samples coming into the small facility, whereas the focused facility, you'd only get cancer samples, brain samples, biomedical tissue, stuff like that. So your sharing policies here is a bit more loose, right? Because you want to push your data out to your clients as quickly as possible. Where from a medical focused facility, you have very strict transfer agreements. Like I had to spend a month writing out data transfer agreements to send data to an institute across the road. So it can be quite bureaucratic. Sometimes each separate facility within one larger institute has their own data sharing policies, which makes things difficult. And large institutes sometimes has an agreement between an institute and a third party. So it's not you sharing to another researcher, it's the institute sharing itself. And the data management is closely linked to staff allocation over the years. And this is where the gross oversimplification comes in. So just allow that for a minute. Where we went from qualitative focused microscopy, where you'd have one researcher and one microscope. With technology development, you'd have adoption of newer technologies, like resolution revolution came in first, SPF I think was in 2004, so everyone's kind of grabbing the newest kind of tech. When we started to think more quantitative, we added a lot more manpower to these larger facilities to manage the processing. But what happened in smaller facilities is you might have added a little bit more compute power, but your manpower stayed the same. And now in the AI era, you have some imaging scientists and a lot of server management going on here, where smaller institutes still have the same amount of manpower sometimes that they had a decade ago. And this causes a little bit of discrepancy in how we help facilities adapt, because we have great policies coming out, and I'm not bashing this at all. But when I looked at this from a small facility point of view, what I saw was something like that, where if you have a lot of amendments to policy and you already have people designated to each part, what happens when you try to get small facilities to this level is it's a little bit daunting trying to catch up. And I think this is what I love about global imaging is we can definitely manage these expectations. An example was when we installed our facilities at Stellenbosch. We had everything ready to go, everything was set up, we had training, but the data management plan was a small NAS behind one microscope. That was it. And we can do a lot better, and we should do a lot better when it comes to infrastructure planning, especially for new facilities. We can definitely simplify this process getting started. A lot of the times people ask, where do I get started? And you can break this down into like 4 steps to know how to navigate. Defining a strategy with your IT department will be the first one. Knowing that you need storage space, so this can be purchased in a small NAS instead of a Miro. I think Gordon showed this brilliantly yesterday with linking their RedCap to a Miro. Defining minimal metadata standards. You can keep it simple but consistent to start with. I think a lot of small apps look at all the metadata standards that are being put out there and they're like, oh, wow, okay, that's a lot. You can keep it very simple to start with. And then additionally, compute power. If you need any additional compute clusters, a lot of, we heard this from Joe now, a lot of countries have national compute resources dedicated to bioinformatics. In South Africa, there was the SKA array and astronomers basically hog the national compute infrastructure. And this is where joining forces with the great bioinformaticians in these countries can really help level the playing field. So in summary, image data management is as much about managing people as it is about managing data. Policies can be adopted globally, but on a more level playing field. The barrier for entry can be minimized, and what I'd like for us to move into is a unified file format and unified repositories to get 2 data sets like this under the same umbrella to upload to the same repository, because at the moment I'm just dumping everything into bioimage archive, even though it's spatial metabolomics. But this is SPF and this is spatial transcriptomics, and they should be on the same file format. So yeah, with that, I know I'm going over time, so thank you.